Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing GAT B entrance exam question paper conducted in the year 2021. I have answered only the biology questions. Then which among the following organs is not capable of secreting hormones? So salivary glands. They do not secrete hormones. Answer is option 2. Then choose the incorrect statement from the following. AUG is initiator codon and also codes for methionine. UGA is a terminator codon. UGA, it is UAG which is a terminator codon, not UGA. So the incorrect statement is option 2. Next one. Which of the following statements about lipid is not true? Glycerol and fatty acids make up lipids. Lipids are water soluble and some even contain phosphorus. Glycerol is highly complex lipid. Glycerol is not a lipid, it is an alcohol. So the incorrect statement is option 3. Then match the components in list 1 with those in list 2. Down's syndrome. Down syndrome is trisomy of 21st chromosome, A4. Then Turner syndrome. Turner syndrome is seen in females only. Phenylketonuria is autosomal recessive trait and sickle cell anemia is point mutation. So the answer is option 2. A4, B3, C1, D2. Which one of the following is a membraneless structure? So centrosome. Centrosome doesn't have a membrane. Cytochrome is found in mitochondria and it is surrounded by mitochondrial membrane. Endosomes also are membrane bound. Peroxisomes are also membrane bound. So the answer here is centrosome. Then next one. Mendel's discovery in pea plants may be considered incidental since the chosen traits were more tightly linked, there was no linkage, only completely dominant, no highly epistatic, no independently assorted. Yes, answer is option 4. Height of a human being is a typical example of which mode of inheritance? So it is an example of polygenic inheritance. And polygenic inheritance refers to the inheritance of a trait governed by more than one gene. That is called as polygenic inheritance. The next one. Which of the following hormones attain the peak of expression in the middle of menstrual cycle? So it is luteinizing hormone. Answer is option 3. Then milk is secreted by the alveoli present in the mammary lobes and it reaches up to the nipple through the following regions. Lactiferous duct, mammary duct, ampulla, mammary tubule. The correct order of the flow of milk. So it is option 2, that is D, B, C, A. D is mammary tubule, then comes mammary duct, then comes ampulla and then comes lactiferous duct. So answer is option 2. Then next one. Given below are two statements. One is labeled as assertion and another is labeled as reason. Most humans pass accurate genetic instruction from one generation to next. That's correct. In spite of high mutation rate, the sequence of the genome changes by only a few nucleotides during division. That is not correct. So in the light of the above statement, choose the correct answer from the options below. So A is true and R is false. Answer is option 3. The respiratory pathway common to erythrocyte and leukocyte is, it is substrate level phosphorylation. That is common to erythrocyte and leukocyte. Then pick the most basic dipeptide in the following. So it is glycine and arginine is more basic than lysine and histidine. So and glutamic acid. So like glycine, arginine is the answer. Then which of the following features distinguishes myoglobin from hemoglobin? Presence of heme prosthetic group, ability to bind to oxygen, molecular size and allostery. Yes, because myoglobin is single subunit and it is tertiary structure. And allostery is found in hemoglobin and it has more than one subunit and it is quaternary structure. So the answer here is option 3. Next one. Match the components of list 1 with those in list 2. Valine, leucine. Valine, leucine are hydrophobic. Serine, threonine are hydroxyl group side chain. Arginine, lysine are basic, and histidine, tryptophan are heterocyclic side chains. So the answer here is option 2, that is A2B4 C1D3. Then next one. Which of the following is not a plant hormone? So brassinosteroid, cytokinin, cytokine. Cytokine are small proteins that are crucial in controlling the growth and activity of other immune system cells and blood cells. So that is not a plant hormone. Cytokine is not a plant hormone, whereas cytokinin is a plant hormone. Then next one. Eclipse period in virus infection is defined as the time. So the period of time between infection by a virus and the appearance of the mature virus within the cell is called as eclipse period. So the answer here is between infection and appearance of mature virus. Option 3. The first virus to be discovered was tobacco mosaic virus. Option 4. Which among the following enzymes enabled integration of the retroviral genome into the host genome? So it is answer is reverse transcriptase. Then several DNA viruses can cause latent infection by their ability to. So viral latency is the when virus is present in the body but exists in the resting, that is latent state, without producing more virus. So they integrate into the host genome but do not replicate. Answer is option 3. A virus X requires endocytosis for infection and replication in the cytosol. Deproteinized genome of the virus is non-infectious. X is enveloped minus strand RNA virus. Answer is option 4. Then which one of the following vaccine was developed by Louis Pasteur? So Louis Pasteur developed rabies vaccine. Answer is option 4. Which one of the following statement about central memory T cells is true? So central memory T cells, they reside in secondary lymphoid organs. Yes, central memory T cells, they reside in secondary lymphoid organs. That is true. So first option itself is true. Here are 4 statements about major histocompatibility complex MHC. So MHC1 is expressed in by nearly all nucleated cells. That's correct. MHC2 is expressed by antigen presenting cells and they present antigen to cytotoxic T cells. Not cytotoxic T cell but to T helper cells. Only helper T cells and not the cytotoxic T cells can recognize antigen in complex with MHC2. That is also correct. Then natural killer cells express a variety of receptors that engage with MHC2. So like T cells, natural killer cells have T cell receptors. Unlike most T cell, the T cell receptors of the natural killer cells interact with MHC like molecules called CD1 rather than with class 1 or class 2 MHC molecules. So out of these, which statements are true? True, A and C only are true. Answer is option 2. Then PSSM stand for? It stands for Position Specific Scoring Matrix. Answer is option 3. Then which one of the following attribute of a macromolecule is modulated by molecular dynamics simulation? So molecular dynamics, it is a computer simulation method for analyzing the physical movements of atoms and molecules. It can be used for ab initio protein structure prediction. So which one of the following is attribute of the macromolecule by molecular dynamics simulation is option 1, that is structure. Then papain digestion of an IgG predominantly results in the formation of. So it results, this is IgG molecule. 1 minute. Okay, this is IgG molecule. It has got disulfide bonds where SS have been marked. This is the heavy chain and this is the light chain. Now upon papain digestion, you will get Fab fragments and FC fragments. So Fab fragments, they have got the ability to bind to antigen. So you get one Fab fragment and you get two Fab fragments and one FC fragment. It is not, you get two Fab fragments, one, two. You get two Fab fragments and one FC fragment upon papain digestion. So the answer here is option 2 Fab fragment, 1 FC fragment. Option 2, 1. Then next one. Which organ is used for isolation of activated B lymphocytes for monoclonal antibody production? It is the spleen. Answer is option 2. The only genetically encoded amino acid without a stereoisomer is glycine. Glycine is the only amino acid which does not have chiral carbon atom. So that is, it doesn't form stereoisomers. So answer is option 3, that is glycine. Then disulfide bonds are identified using the distance between the side chains of which of the following sulfur containing residues? So that is cysteine and cysteine. Answer is option 2. The two strands in double helical DNA are held together by hydrogen bonds between nitrogenous bases. Answer is option 4. Which one of the following is a receptor for HIV-1? So T helper CD4, that is T helper cells are attacked by HIV-1. So CD4 is the answer. Then next one. A student carried out expression analysis of a gene P10 in a cancer cell line using both northern blotting and western blotting. He observed a decrease in the western blotting signal but no change in northern blotting signal compared to normal cell. In this context, which of the following statement is correct? Now the student carried out expression analysis of the gene P10 in a cancer cell line. He used both northern blotting and western blotting. And what did he observe? He observed there was a decrease in the signal in western blotting, but in case of northern blotting, western blotting is for protein studies and northern blotting is for study of RNAs. So he saw that there was a decrease in the signal intensity with western blotting, but there was no change in the normal blotting compared to the normal cell. In this context, which one of the following statement is correct? So answer here will be option 2. There is a decrease in P10 protein level but no change in the P10 mRNA expression in the cancer cell. So answer will be option 2. Which one of the following DNA sequences can act as genetic markers? So they are long terminal repeats, LTRs. Answer is option 4. Then which of the following is not a genetic disorder? Cystic fibrosis is a genetic disorder. Sickle cell anemia is also a genetic disorder. Down syndrome is also a genetic disorder. Marasmus is not a genetic disorder. Then zoonotic viruses are those that can be transmitted. So zoonotic viruses can be transmitted from animals to humans. Answer is option 3. Given the molecular weight of glycine is 75, what would be the molecular weight of glycine-glycine dipeptide? Now each peptide bond, it removes one molecule of water. So dipeptide means 75 into 2. So that will be around 150. And from this 150 you have to minus 18. 18 is the water molecule that is removed. So the molecular weight decreases by 18. So 150 minus 18 will be 132. So answer will be option 3. Then next one. Given 3 samples, L-tyrosine, D-tyrosine, equimolar mixture of L and D-tyrosine. Which one of the following methods can help in the identification of these samples? So now The L and D forms are said to be optically active and they rotate the plane polarized light in different directions. So you can use optical rotation method for identification of these samples. Answer is option 4. Now identify the form of chromatography, reverse phase or gel filtration as shown in the chromatograms A and B below. So in the case of A, salt is eluted first, then comes your protein, and in the case of B, protein is eluted first, then comes salt. Now if you see the case of reverse phase chromatography, it employs a polar mobile phase. As a result, hydrophobic molecules in the polar mobile phase tend to adsorb to the hydrophobic stationary phase, and hydrophilic molecules in the mobile phase will pass through the column and are eluted first. So salt is hydrophilic and it will be eluted first. It will not bind to the column since it is not hydrophobic. So the first one will be reverse phase chromatography. And the next one, gel filtration chromatography, it employs porous beads with a defined pore size distribution as the stationary phase. Small molecules can enter the pore and hence elute last, whereas large molecules they are will elute in the beginning itself. That is, they will elute first because they will not be able to enter the pores. So salts are small compounds and they will enter the matrix and will elute last. So the answer here is first one is reverse phase and the second one is gel filtration chromatography. Then how much will 1 mole of glucose weigh in grams on the earth and on the moon? So 1 mole of glucose, that is molecular mass of C6H12O6 in grams, it is 180 grams. So chemical analysis, it is always based on mass, so the result will not depend on the location where you are weighing it. So the answer will be, it will be 180 on both earth and on the moon. Then what are the number of moles in 100 ml water and 100 ml heavy water respectively? So if you see water, this is the molecular weight, that is 18, and heavy water it is 20. Now mass is equal to volume into density. They have given the density values. So for normal water it is 100 grams and for heavy water it is 111 grams. And now calculate the number of moles. So weight by molar mass, that is 100 by 18, that is 5.55 moles. And for heavy water 111 by 20, that is also 5.55 moles. So the answer will be option 1. Then from the table given below, what is the overall protein yield? So this is the purification table. They have crude extract, then they passed it, then they did ammonium sulfate precipitation, then they passed it through ion exchange chromatography, then size exclusion chromatography and affinity chromatography. This is the yield. Now they have asked for overall protein yield. So that will be multiply everything by 100. Sorry, multiply all the answers. So 100 into 96 by 100 into 80 by 100 into 60 by 100 into 45 into 100. So that will be 20.70%. Answer is option 2. Coming to the next question. Calculate the approximate concentration of a solution with molar extension coefficient 650 molar per centimeter, path length 1 centimeter and absorbance 1. So you know the formula A is equal to epsilon Cl. Substitute the values into the equation and calculate for C. C is equal to A by epsilon L. So the answer here will be 1.5 millimolar. Answer is option 1. A 5200 base pair long cloning vector contains two BAM-H1 sites that are 450 base pair apart. In a cloning experiment, a 1200 base pair long gene X having two BAM-H1 sites at 3' and 5' end was PCR amplified and cloned after complete digestion of the vector with BAM-H1, what will be the size of the resultant plasmid containing the cloned gene X? Now, this is the plasmid that is 5200 base pair and it has got two BAM-H1 sites that are 450 base pair apart. This is the gene of interest that is 1200 base pair and it has got two BAM-H1 site at 3' and 5' end. And this region was amplified by PCR and cloned after complete digestion of the vector with BAM-H1. So the vector was first digested with BAM-H1. So this 450 base pair was removed and in place of that this 1200 base pair was added. And then you got a resultant plasmid with the gene of interest or containing the gene X. So now what they have asked, what will be the size of the resultant plasmid containing the cloned gene X? Now vector, if you remove 450 base pair, what will be remaining size? 4750 base pair. Now after cloning with 1200, what will be the remaining size of the plasmid? So 4750 plus 1200, you get 5950 base pair. So the answer here will be option 3. Match the various nucleases in the list 1 to their respective function in list 2. So DNase 1, it degrades both single and double stranded DNA. RNase H, it degrades RNA DNA hybrid. RNase A, it degrades single stranded and double stranded RNA. And S1 nuclease, it degrades single stranded DNA and RNA. So the answer here is option 4. A3B2C4D1. Answer is option 4. The difference between a cosmid and a plasmid is the presence of which of the following additional features in the cosmid. So cosmids are plasmids containing more than one or more than one cohesive sites or cos sites of the bacteriophage lambda. So lambda-phage DNA sequence that includes a cos site. Answer is option 1. Nick translation is carried out by DNA polymerase 1. A single cell can divide and give rise to completely differentiated organism is referred to as, it is called as totipotent. Plant cells are totipotent. Okay. So what is pluripotent? Pluripotent is capable of giving rise to several different cell types. Unipotent, capable of giving rise to only one cell type. And multipotent, they have the capacity to self-renew by developing and dividing into multiple specialized cell types present in a specific tissue or organ. So the answer here is that give rise to a completely differentiated organism and it is referred to as totipotent. Then which of the following statements are correct? Mitochondria and chloroplast contain DNA and are associated with ribosome along with enzymatic machinery of protein synthesis. That is correct. Mitochondria and chloroplast contain DNA but do not make protein. They make protein. So option B is not correct. Mitochondria and chloroplast DNA sequence or ribosomes present are more related to bacteria and cyanobacteria than to eukaryote. That is also correct. Mitochondria and chloroplast are distinct entities and can survive outside the cell. That is not correct. Neither mitochondria nor chloroplast contain all the genes necessary to specify all the proteins present in these organelles. That is also correct. They require some nuclear proteins for their function. So they synthesize their own proteins, but they also require nuclear proteins for their function, proper function. So the answer here is A, C and D. Option A is the answer. Then next one. An organelle found in cytoplasm of eukaryotic cells containing oxidative enzymes capable of energy generation is. So it is peroxisome. They contain oxidative enzymes such as catalase and urate oxidase. Then which one of the following statement is not true about the role of primase enzyme in DNA replication? It does not need a primer to function. That itself is wrong. So option 1 is the answer. During evolution, mitochondria have been acquired from endosymbiotic bacteria. Answer is option 3. Which of the following statement is not true about adjuvants? Adjuvants are substances that when mixed with an antigen and injected with it enhances the immunogenicity of the adjuvant. So it creates covalent modifications over the epitope. That is not correct about adjuvant. Answer is option 3. Which amino acid can be quantified from acid hydrolates of protein? So, acid hydrolysis is with 6 molar HCl yielding the desired individual amino acid. However, not all the amino acids can be completely recovered upon HCl hydrolysis or acid hydrolysis. So some amino acids are hydrolyzed to their acidic form such as arginine and glutamine to form aspartic acid and glutamic acid respectively. So you cannot quantify arginine and glutamine. Then you cannot differentiate between that is you cannot differentiate between glutamic acid and glutamine, similarly between aspartic acid and aspargine, so they cannot be quantified. So the answer here is which amino acids can be quantified from acid hydrolates of protein is histidine. Answer is option 3. Then next one. The molecular formula for glycine is C2H5O2N. What would be the molecular formula for a linear oligomer made by linking 10 glycine molecules together by condensation synthesis? So formula for glycine is C2H5O2N. Now linking 10 glycine molecules, then multiply by 10. That is 2 into 10, 5 into 10, 2 into 10, 1 into 10. Now there will be 9 peptide bonds between glycine residues, and through the bond formation one molecule of water is removed. So now you have to subtract 18 water molecules. So 9 into 2 and 9 into 1. Sorry, not 18 water molecules, 9 water molecules. Okay, my mistake. Subtract 9 water molecules. So you will get C20, H32, O11, N10. Answer is option 2. Then, based on phosphoryl transfer potential, identify which one of the following cannot help in the generation of ATP. So glucose 1-phosphate, it is a phosphoester bond and they are not high energy rich bond. So glucose 1-phosphate is the answer. Then the Michelson and Stahl experiment showed the DNA replication is semi-conservative. Answer is option 3. The primary enzyme responsible for direct reversal of UV-induced thymine dimers are photolysis. Answer is option 2. Post-translational phosphorylation of protein usually modify the serine, threonine and tyrosine residues. Then next one, aqueous organic solvents will have. So higher the polarity, higher is the dielectric constant. So aqueous organic solvents will have they have asked for organic solvents, and organic solvents are non-polar, so they will have lower ionizing capacity than pure water. Answer is option 1. Then phenyl isothiocyanate efficiently reacts with amino groups of proteins. So phenyl isothiocyanate is reacted with an uncharged N-terminal amino group under mildly alkaline condition to form a cyclical phenyl thiocarbomoyl derivative. So under alkaline conditions, answer is option 4. Which class of neurotransmitters would be most affected by a toxin that disrupts microtubules within neurons? So peptide neurotransmitters are produced in cell body and are transported to the axon terminal. The peptide neurotransmitters transported via the microtubules. If the microtubules are disrupted, then the neurotransmitter will not be able to be transported. So the toxin that disrupts microtubule within neuron, the class of neurotransmitter that is most affected is peptide neurotransmitters. Answer is option 4. Then which of these is a disease of the myelin sheath? That is multiple sclerosis. Answer is option 3. Which of these techniques cannot differentiate between identical twins? So DNA fingerprinting cannot be used for distinguishing between identical twins, because identical twins the genome is 100% similar. So DNA fingerprinting cannot be used. Then next one. Which of the following is not a component of plasma membrane? That is histones. Answer is option 2. A solid which is not transparent to visible light and whose conductivity increases with temperature is formed by covalent bonding. Answer is option 1. Next one. In transmission electron microscope, a beam of electrons interact with the specimen to form the image as a shadow. Answer is option 2. Then next one. The concentration of ligand is equal to Kd, that is dissociation constant. When the fraction of the ligand bound to the protein is. Now if the ligand concentration is equal to Kd, then half of the protein is bound to the ligand. So the answer here is option 3. The specific cleavage at phenylalanine tyrosine tryptophan amino acid is catalyzed by. So it is catalyzed by chymotrypsin. It cleaves at C-terminal of phenylalanine, tyrosine and tryptophan. Then D-alanine and L-alanine are technically known as enantiomers. Enantiomers are pair of molecules that exist in two forms that are mirror images of one another but cannot be superimposed one upon the other. As in the case of D-alanine and L-alanine, they are mirror images. So answer will be option 2, that is enantiomers. Then in the lac operon, promoter and operator refer to. Okay, promoter is the site where RNA polymerase bind and operator is bound will be bound by the repressor. So the answer is option 2, binding site for RNA polymerase and repressor. The bacterial mRNA binds to ribosome through the, so it is the Shine-Dalgarno sequence. It is a sequence of 5 to 9, or you can say approximately 7 nucleotides located upstream of the start codon in prokaryotic mRNA that is recognized by the ribosome. Then EcoR1 enzyme recognizes the sequence GAATTC. A stretch of linear DNA with 6 sites will give rise to. So you have got a DNA like this and it has got 6 sides. 1, 2, 3, 4, 5 and 6. So how many fragments will you get? 1 minute. So there are 6 sides. 1, 2, 3, 4, 5 and 6. How many fragments you will get? 1, 2, 3, 4, 5, 6 and 7. So the answer here is option 2, that is 7 fragments. Which of the following activities is associated with reverse transcriptase enzyme? So RNA dependent DNA polymerase. RNA dependent DNA polymerase is a DNA polymerase enzyme that transcribes single stranded RNA to DNA. So that is associated with reverse transcriptase enzyme. Then what is the minimum number of beta strands that can be twisted and packed to form a beta barrel structure? So the answer is option 3, that is 8. In sickle cell anemia, the negatively charged glutamic acid residue is replaced by which one of the following neutral amino acids? So glutamic acid replaced by valine. Answer is option 2. At what level of structure do myoglobin and hemoglobin differ with the most with respect to their function? So they differ the quaternary structure. Myoglobin is tertiary structure, hemoglobin is quaternary structure. The next one, hemoglobin's P50 value is about dash as great as myoglobin's P50 value. So the P50 is the oxygen tension at which hemoglobin is 50% saturated. The P50 of hemoglobin is nearly 10 times higher than that of myoglobin. So the answer is option 3. Alpha D glucose and beta D glucose, they are anomers. Answer is option 4. The major storage form of lipid is triacyl glycerol. Answer is 4. The dihedral angle phi and psi are referred to as Ramachandran angles. 2. The molarity of pure water is 55.55. I have solved it in the next slide. You can go through it. This question is even asked for the NEET exam also. It is seen in most of the entrance examination question paper. What is the molarity of pure water? So here is the explanation for molarity of pure water. You can go through it. Okay, an oxidizing agent is defined as, so an oxidizing agent, it is a substance in a redox chemical reaction that gains or accepts or receives an electron from a reducing agent. So it is an electron acceptor. Answer is option 2. Then emulsifier is an agent which stabilizes the emulsion. Answer is option 3. The metal atom present in vitamin B12 is, so it is cobalt. Answer is option 1. Fleming's left hand rule is associated with the effect of magnetic field on current. Answer is option 4. Match the components of list 1 with those in list 2. So cooperative binding of oxygen by hemoglobin, Archibald Hill. 4. A4. Production of monoclonal antibodies, Caesar Milston. B1. X-ray diffraction pattern of myoglobin, John Kendrew. Formation of enzyme substrate complex in catalysis, Victor Henry. So the answer here is option 4. Then next one. The restriction endonuclease which cleaves the given sequence at the indicated site is, so the sequence is GGATCC and that is the sequence of BAMH1. Answer is option 2. Choose the correct statement about kelps. It is a freshwater alga. It is not freshwater, it is marine alga. It attains red color due to the presence of phycoerythrin. It is a brown alga, so it is not going to be, not a red alga to attain red color. Then it is a marine weed possessing fucoxanthin and carotenoids. That is correct. The cell wall of kelps contain alginine and pectin. It does possess cell wall made of cellulose and alginic acid. So the answer is option 3. The term used to describe genes arranged in the same order on chromosome of different species is called synteny. Answer is option 4. Then aroma in rice is due to which of the following compounds? So it is 2-acetyl 1-pyrroline. Answer is option 2. Then which one of the following techniques can be used to overcome pre-fertilization barrier between two plant species? So it is protoplast fusion. Answer is option 2. A man with AB blood group marries a woman with blood group O. What is the probability of having O blood group among the offspring? So this is the AB blood group man. He marries a woman with blood group O. So they get offsprings AO and BO. So A and B will be the blood groups. No O blood group children will be born to them. So no offspring will have O blood group. Answer is option 4. Then next one. An unknown solution shows distinct peaks at 260 and 280 nanometer in a UV spectrophotometric analysis. The solution may contain proteins, DNA and RNA, because nucleic acids, that is DNA and RNA, will give a peak at 260 nanometers and proteins will give a peak at 280 nanometers. So answer is option 2. Now, which of the following subcellular structure are not present inside the nucleus? So, it is Parr speckles. See, 1 minute. Parr's speckle is an irregularly shaped compartment of the cell found in the nucleus interchromatin space. So, they have asked which is not present inside the nucleus. So Golgi body is not present in the nucleus. Mitochondria is not present in the nucleus. Endoplasmic reticulum is also not present in the nucleus. So B, D, E. Answer is option 2. Then a promoter is. So a promoter is a sequence of DNA recognized by RNA polymerase to initiate transcription. Answer is option 1. A ribose switch is. So a ribose switch is a regulatory segment of an mRNA molecule that binds a small molecule, resulting in a change in the production of the protein encoded by the mRNA. So the answer here is option 2, a structured regulatory mRNA segment that binds a small molecule. Then next one. One of the limitation of Y chromosome DNA profiling is that, so Y chromosome, it usually comes from the father. There is no Y chromosome in the mother. So the chances of recombination in Y chromosome is not there. Whichever Y chromosome the father has, it will go to the son and to other male siblings. So we cannot differentiate which Y chromosome is from father or son or from the male siblings. So the answer is option 2, that is it cannot differentiate between DNA from father, son and male siblings. Answer is option 2. Then next one. Match the enzymes in the list 1 to their respective functions in list 2. DNA primase. Synthesis of RNA primer during replication. RNA polymerase, mRNA synthesis during transcription, reverse transcriptase, complementary DNA synthesis from RNA, poly A polymerase, 3' end addition of adenosine nucleotide. So the answer here is A3B4 C2D1. Then next one. Determine the sequence of genes on a chromosome based on the recombination frequencies given below. Mn 10 map units, Mo 26 map units, Mp 20 map units, No 16 map units, Np 38 map units. So if you have the sequence Mp, Mno, you can see that P and M or M and P are 20 map units, M and N are 10 map units, N and O are 16 map units, and MO is 26, 10 plus 16, 26 map units, and NP, that is 38 map units. And one more thing I would like to tell is that these they are not additive. So linkage maps are not additive. So always 10 plus 16 will need not be 26. Here 20 plus 10 is 30. It is not 38 as they have given in the question. If it was additive, it would have been 30, but it is not additive, so that is why P to N is 38. So answer here will be PMNO. Answer is option 2. Then next one. Evolution of life forms had occurred due to use and disuse of organs. This theory was postulated by Jean-Baptiste Lamarck. Answer is option 1. Which of the following features is true about all 20 genetically coded amino acids? They are optically active. Not all are 20 are optically active. Glycine is not optically active. So is true, they have asked. They contain a methylene group in their side chain. That is also not correct. They contain carbon, hydrogen, oxygen, nitrogen. That is correct. Answer is option 3. They are basic in nature. All are not basic. Some are acidic also. Then next one. A DNA sequence has two ends, 5' and 3'. Which of the following statement is correct regarding the nature of the ends? So the 5' end it contains the free phosphate group and 3' end it has got the free hydroxyl group. So the answer here is the 5' end has a phosphate group. Answer is option 2. The next one, telomerase enzyme is. So telomerase is the enzyme required for the addition of telomeric repeats to the ends of a linear chromosome. And it is a reverse transcriptase. It is RNA dependent DNA polymerase, meaning the enzyme that can make DNA using RNA as template. So it is RNA dependent DNA polymerase. RNA dependent or reverse transcriptase. Answer is option 4. Then which of the following is true for single cell protein? Algae cannot be used in single cell protein. Algae is used in single cell proteins. It is produced through fermentation. Yes, that is correct. It does not contain carbohydrates and vitamin. It does not contain carbohydrate, but it is a good source of vitamin. Its utilization increases environmental pollution. It doesn't increase environmental pollution. Answer is option 2. The Cartagena Protocol regulates trade in. So it is for genetically modified organisms. The Cartagena Protocol regulates trade in genetically modified organisms. Then given below are two statements. One is labeled as assertion and the other is labeled as reason. So assertion, biodiversity of the prokaryotes cannot be quantified precisely. Reason, conventional taxonomic methods are not suitable for the identification and characterization of all microbes. In the light of the above statement, choose the most appropriate answer from the options given below. So both A and R are correct and R is the correct explanation for A. Answer is option 1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.